This is TWIS. This Week in Science, episode number 783, recorded on Wednesday, July 22nd, 2020. Why the Earth isn't flat. Hey everyone, I'm Dr. Kiki, and tonight on This Week in Science, we will fill your heads with nanostars, invasive hedgehogs, and brains. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. I'm about to give you some advice. The following advice is just a bunch of words being said by someone who very rarely gives good advice, let alone takes any. And yet somehow reigns a desire to, above all else, not look like an idiot. At least not in public, and especially not on a video recorded format like this, where people can watch you be an idiot over and over again by replaying idiotic things that you said. Believe me, I have done it. I know there are clips available of me saying the darndest things, and so I have compiled a list. Simple list, 3 things you can do to avoid looking like an idiot in public. Number 1, know what you're talking about. Know at least the basic facts, and more is better. It may require reading or even listening to others, but the more you know about a thing that you are talking about, the less of an idiot you will seem. Number 2, if you say some stuff that you might not pretty sure definitely isn't true, don't ask to be fact-checked on the spot, unless you genuinely, honestly want to know the truth. C, when faced with facts that you yourself asked for, which refute the thing that you previously said, remember that most people watching are not in your payroll, they're not your family, and if you stick to the now proven very wrong thing that you said, they will openly and mercilessly mock you for being an idiot. And 4, do not base the credibility of your intellect on a test designed to look for cognitive impairment. Smart people never brag about how smart they are, especially after taking a cognitive impairment test. That's what idiots do. And lastly, remember, while not being an idiot isn't everything, it is something you are able to achieve simply by listening to This Week in Science. Coming up next.
I've got the kind of mind I can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
Good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back again. Yes, we are, to talk about science. Science. That's right. We're here to talk about science. I hope that's why you are listening to this podcast right now, is that you're interested in science, or you hope to be. We hope to fill your minds with wonder at what the heck we're doing here. No, wonder about the world around you. That's right. I have stories tonight about batting 1,000. Yes, batting 1,000, and a baby picture, a little tiny baby picture, and brains, because brains are my thing. I like them. Justin, what did you bring?
I've got silver-coated golden nanostars, breaking a record in 7th place, oldest evidence of Native Americans, just got a whole lot older or a little bit older, depending on who you ask, and why plants are a good thing to have.
Because of oxygen? Yeah. I mean,
another good reason for plants.
Oh, okay. Right. All right. More good reasons to love things. Love the
plants.
Green things. Yes. Blair, how's that animal corner looking tonight? What do you have for us?
Well, I have pigs, brains, toxic aggression, and invasive hedgehogs.
Oh.
Those invasive hedgehogs, I think that is, I kind of want to name a band, Invasive Hedgehogs. The Invasive Hedgehogs. It'll be I don't know, metal band? I'm not sure.
Sounds, yeah, and they would wear quills everywhere, I guess. Yeah.
Yeah. All right. Are we ready to jump into the show tonight?
Let's do it.
Let's do it. Okay. I just want to remind everyone as we dive on in that if you are not yet subscribed to This Week in Science, you can find us in all of the podcast players: Stitcher, Spreaker, TuneIn, Apple, Google, radio.com. Look for This Week in Science and you will be able to find us. We are also on YouTube and Facebook, and our website is TWIS, TWIS.org. Okay, let's dive into the science. Quick science stories. Let's start off batting 1,000 and set the trend for the show. Well, batting 1,000, I'm not talking about bats. I'm talking about bats. Not baseball, but bats. The
1,000
animal.
of them?
The flying mammals that are actually related to whales. That's, yeah.
Yes. And it's so surprising about the whales.
And pangolins.
People like to say that they're related to mice, and that's their closest relative, but no. Oh, no, no, no. Researchers from the BAT-1K, the BAT-1000 project, have sequenced the genomes of 6 bat species. And they compared these sequences with many, many, many other animals. The results suggest that bats as a group might share a common ancestor somewhere back a ways with horses, whales, pangolins, the aforementioned animals, and dogs. And that they have always had an amazing immune system. That their immune system isn't just good now, but it's always been good. Digging into their genome, they find all sorts of leftover bits of viruses from long ago infections that indicate that they were, at one point in time, infected by viruses that are thought only to attack birds. So bats get attacked by all kinds of things and are fighting them off. And so they've always had this really strong immune system. Additionally, they have discovered that within the genome, there are lots and lots of replications of antiviral genes. So while they have turned off about 10 genes that are shared with other mammals for the fight against viruses, which you would think, oh, those genes are turned off. They shouldn't have a strong immune system. There are a number of other genes that are very important to fighting viruses that are replicated and that have unique mutations. So looking into those a little bit more in the future might lead us to some interesting insights into exactly how the bat immune system works now and has worked throughout history. The researchers hope that they'll be able to sequence 27 more bat species genomes next year. They're currently applying for funding. 6 this year, 27 next year. I mean, they're just going for it. It's batty.
I mean, I guess that's kind of a weird, I don't know if I would even call it a bright side, but I guess it's something that's happening as a result of the kind of the arrival of COVID is bats and pangolins are getting some research focus that they didn't have before.
Yeah. I hope that the pangolins get a lot more research. I mean, bats, especially because there are so many species that transfer, they harbor diseases. They're vectors, known vectors. I mean, this is a really important area of research.
So aside from being, sort of vectors now, it sounds like they've always been like a place viruses like to hang out in bats, which might also have led to some of this immunity, so maybe, that's what you want to study. The thing that gets the most viruses and doesn't die from them. Perfect.
Yes, exactly. Now tell me about some of these nanostars.
Okay. So this is from, these are inverse molecular sentinels. This is the actual official sciencey name for them. These biomedical engineers at Duke University engineered a method for detecting the presence of microRNAs. Very specific ones. They have to sort of know what they're looking for in advance. So they can just take a sample tissue and pull from there it says here sort of without the need for labeling or target amplification. You don't really need to do the amplification, but there is some labeling involved on this. After all, the technique that they use to identify early biomarkers of cancer and other diseases without the need for time-consuming, expensive processes, special laboratory equipment. MicroRNAs are these short RNA molecules that bind to messenger RNA and stop them from delivering sections of DNA into the machinery that does the replication. So in doing this, microRNAs regulate, modulate, and even silence certain sections of DNA or gene expression, which then alters the behavior of certain biological functions, because now things that could have been printed aren't being printed because the microRNA said, nope, you're not going to work today. But the general focus of this is Juan Vodin, professor of biomedical engineering at Duke, who says, the general research focus of my lab has been on the early detection of diseases in people before they even know that they are sick. And to do that, we'll need to be able to go upstream at the genetic level, genomic level, and look at the biomarkers for these diseases, such as microRNA. So Professor Vodin designed a faster, better way to identify specific microRNA. Gold nanostars, this is the thing he's created. These gold nanostars have multiple spikes that can act as lightning rods for enhancing electromagnetic waves, which is a unique feature of the particle's shape, says Vodin. Our tiny nanosensors, called inverse molecular sentinels, take advantage of this ability to create clear signals of presence of multiple microRNAs. The strategy of these things is to get a little molecular label near the pointy parts. And when the specific stretch of RNA attaches to this label, they shine a laser and it kind of arcs. Under laser excitation, the label emits a light called a Raman signal, which is generally very weak, but the shape of the nanostars, coupling effect of separate reactions caused by the gold nanostars and their silver coating, amplifies signals several million folds, making them easier to detect. And what he's finding is basically they can put these in with the samples and it's going to basically come back with a spike on the computer. And these, he's saying is like a fingerprint of the specific differentiated microRNAs. By being able to spot which microRNAs are present, they can then predict what disease will follow from the presence and the number that they're seeing there. Really amazing, incredible feat, having a sort of lab-proof method.
That's amazing, yeah.
Absolutely will accelerate that field.
Yeah, anything that makes the methodology more robust, then it can be replicated more places. It can work in a larger number of lab environments. You know, once you get that methodology, it's like, okay, it'll work. It's going to be effective. Super important. But, you know, sometimes some things are less important than others. No, less dependent than others. Blair, what's your next story?
Well, researchers wanted to know why dogs are so gosh darn special. And we know that dogs look at humans in a problem-solving context to establish attention and initiate communication interactions. And that's from an early age. Puppies do it, right? But they wanted to know if this was dogs specifically, or would other companion animals do the same? And of course, we know cats don't. That's probably because they're not social. We know that wolves don't. It's probably because they're not domesticated. So what about pigs? They're domesticated. They can be raised as family pets, and they're also very smart. So
George Clooney has a pig.
Yeah, and I knew someone growing up who had a pig and knew how to roll over and spin in a circle and shake. And yeah, so they're actually really smart and really trainable.
And could their pig do any tricks?
Very good. Anyway, the question was, would pigs look to humans the same way that dogs do, raised in the same situation? And so first they just checked what they did in a neutral situation, which is just, you know, they were in a room and there was food in a dish, basically. And both the pigs and the dogs interacted with the humans equally. They sought out attention from humans because they were raised around humans. But where it changed was the problem solving. So when they made it harder for those pigs to get their food from the dish and the dogs gave up pretty much right away and went to the human to ask for help. But the pigs stayed at it. They remained independent and they continued to figure out how to get the food on their own. Now, it was a trick. The case was locked, so they couldn't get the stuff. But it was supposed to make it simulate being hard, and then the human could open it for them. But yeah, the pigs appear to be more independent.
You know what I immediately think though? That they assume that they're smarter than the humans. I could ask, what would they know?
Probably.
I was assuming the dogs know that we are trained to help them.
Yes.
And that really is the difference. They've been training us since day one. Maybe pigs aren't training us as well.
Yeah, I think our relationship with pigs is quite different. Even though pigs can be trained and they're domesticated, the relationship is not the same.
Mm-hmm. And that's what this boils down to. The relationship is not the same. That's exactly what they were trying to test.
Yeah. Yeah. And it has, who knows, maybe someday it will be.
I mean, it could be. They do make pretty good pets. They're very clean.
And they're
smart.
Cute.
They're trainable.
They're smart.
There's a therapy pig at SFO that you can pet and stuff that they walk around with.
Really?
Yes.
I've never seen the SFO therapy pig.
Yes. I have not seen her in person, but I've read articles about her. And every time I take a flight, I'm like, is she here? Is she here?
I want to hang out with the SFO therapy pig.
Yeah. I think it might be. You might have to have something traumatic to get to meet the pig.
No, it's just to calm people down because people get stressed out before flights. So this is a normal thing. Dogs go to airports all the time as like therapy dogs to help people just de-stress before they get on the plane. But there's a pig at SFO. Fun fact.
So that dog who's sniffing my luggage with the officer.
No, that's a dog with a different job. So the's different jobs that dogs can have in an airport. It turns out. Don't pigeonhole them.
Oh, I wonder what dogs and pigs dream of when they're little puppies and piglets. What are they going to be when they grow up? Yeah.
Well fed, I think.
Does a small puppy dream of being a luggage bomb detecting dog?
I think they dream of treats and sniffing other dogs' butts. That's what I think they dream about.
Yeah.
So there's something to this, Kiki, because if you dogs that have been bred and used as work dogs for many years will really early on start to exhibit some of the behaviors of what they've sort of been bred to train for. There's a really, if you look at a little baby border collie, they will start trying to herd things right away.
Right away, yeah.
My corgi definitely has always she doesn't nip, but she like pushes her nose into the back of our heels. Like she's herding us.
Keep moving.
Where are you going?
Where are you going? I gotta herd you.
Yeah.
Oh, well, after you've heard this story, you might just need that therapy pig.
Oh no.
A new analysis of climate sensitivity.
Oh, no.
The measure of just how much the climate is going to heat up with a doubling of carbon dioxide in the atmosphere updated the previous estimate that was calculated back in 1979, weaving together three separate lines of evidence. An international team of climate scientists produced a 66% confidence interval, and confidence intervals are the range of numbers within which you are fairly certain something will happen. 66% is 66% of the time the numbers will fall within this range. And that range was between 2.6 to 3.9 degrees Celsius in warming. They also, and the reason they did a 66% confidence interval is because that is the range that has been reported by the IPCC historically. And so they wanted to match that. They also reported a 90% confidence interval, which extends the range of numbers to get 90% of the values into that interval. And the range is 2.3 to 4.7 degrees Celsius. So what does this mean? It basically means that no matter how hard we work to reduce that carbon dioxide output, we are not going to be able to keep warming below 2 degrees Celsius. that ship has sailed. We are well into the 2 to 2.5 at the minimum end of warming for the amount of warming globally. And this is not local warming, but global average increase in temperature. And of course, being degrees Celsius, if you were to transfer that to Fahrenheit, that's about 7 degrees Fahrenheit increase globally at minimum.
That's a lot. That's like a lot. That's a lot.
Yeah, let's keep it in Celsius. It sounds better.
I know, keep it in Celsius, people.
Goodness.
I don't even want to bring up the fact that we're still accelerating down this thing. I mean, COVID's been helping, but so when we're talking about 7 degrees, it's probably going to be 20 before we get.
Fingers crossed that it is not, and we can hope that in the next few years we can turn some things around. I am optimistic. We saw a lot of global adoption of behaviors by individuals that managed to shift the curve of the pandemic, and that was not necessarily with it was with government recommendations, not necessarily with great government oversight all the time. But individuals working together are able to enact change. And
it's
that's
about,
where I take optimism.
Yeah, it's about urgency, it's about agency, and it's about community.
Yeah.
And those are the things that have really been successful with COVID, right? Is there's this thing that's happening, it urgently needs our attention. That is not necessarily been part of the climate narrative, so that's something we can work on. People think it's a far off, not today problem, and it certainly is. Take care of your neighbors, take care of the first responders, take care of each other. And the agency, there were very simple things that you can do to make an actionable big difference. And so I think even though it's not perfect, like you said, we are seeing entire shifts in the way we live our lives in a lot of cases. So I think there's something to be learned here about how to move forward with climate.
I think so. I definitely think so. Keeping to the rundown of the quick stories.
No, I've got one still. You're just skipping my story. I got one left.
Yeah.
Congratulations to Tropical Storm Gonzalo. It is now the earliest arriving number 7 named tropical storm on record in the Atlantic Basin, beating out Tropical Storm Gert of 2005 by 2 days. If you don't remember Tropical Storm Gert, that's okay. It never became a full-fledged hurricane, just a big storm at sea. However, not for nothing and worth noting that 2005 was the year Hurricane Katrina struck the States. Not that there's any correlation, but regardless, hurricane season is here. And so if you live in a hurricane-frequented location, maybe it's a good time to get your emergency get-out-of-town kit put together. Gonzalo is expected to go full-fledged hurricane over the next few days, which is just doing the maths here. Hey, I think that will make it the earliest forming number 7 named tropical storm to go hurricane in recorded history as well. So nicely done, Gonzalo. You really did it.
Yeah.
Now,
Follows
please
on the heating very, very easily.
Now, please leave quietly via sea stage, right? Also, I'm pointing this out. NASA's satellite provided the look at the early record breaker, proving that despite pressure from some politicians who want NASA only to look out into space, NASA does a pretty darn good job of monitoring conditions down here on Earth as well.
Yeah. It is one of NASA's essential services to the people here. Faux show. Man, the storms keep getting earlier, bigger. Good job, Gonzalo. Hey, Blair, what's your next story?
Oh, I say potato, you say potato. I say mouse brain, you say human brain. What's the difference?
What?
This is a piece of research out of Tel Aviv University.
Quite a few differences. I don't know where to start.
Well, I'm here to tell you there's not a lot. So this is a first of its kind study looking at brain connectivity in 130 mammalian species. This is a
versus
huge
a potato.
Study. No. So all the brains were removed from dead animals except for the 32 living humans who were also scanned in an MRI. All the animals that had their brains removed were already dead. They were not euthanized for the study. But we were able to see, the researchers were able to see this like huge, vast look at brain structure for 130 different species, from tiny bats again weighing 10 grams, to dolphins in the hundreds of kilograms. So what they found was that the efficiency of information transfer through the neural network does not depend on size or structure, and that they exhibit equal connectivity and information travels with the same efficiency within each brain. They also all had the same balance to preserve that connectivity and information transfer via a special compensation mechanism. So when connectivity between hemispheres is high, it's within each hemisphere, the connectivity is low, and vice versa. So they have this compensation that they do to make sure that they're retaining this efficiency. So, of course, many scientists have assumed that connectivity in the human brain is higher than other animals. But as listeners to this show are probably not so surprised to hear, nah.
No.
Not a brain's a brain.
Yeah, I love one by one these ideas about how the human brain, it's this amazing thing that's better than all the other brains.
We've got to be special.
I mean, we are special, but but it's like, every parent's child is special as well.
I think it's like the what makes us special is not our structure. I think that's where this differs. It's we weren't handed a different set of equipment.
No,
we are mammals. We are primates. We are vertebrates. We have the same equipment as everybody else. It has grown or adapted or evolved to be used in different ways potentially, but we started with the same building blocks.
And I think that's the Legos are the same. The properties of the building blocks are the same, whatever mammalian brain you're looking at. But when it comes to, like you say, the structure, yes, human brain, we have more specialized areas of the brain for things like language, different aspects of cognition. We have maybe different connectivity in the sense of what exactly is connected to what, but in terms of information flow, all the stuff that allows that to happen is the same.
I'm kind of confused by it. Is this just saying that like the speed at which a neuron travels is like the same, or is it saying there's the same number of connections? Like the white matter is no more thick or dense or...
So they are looking at white matter in the brain so that they could reconstruct the neural network. They reconstructed the neurons and the axons, the nerve fibers through which information is transferred, and the synapses where they meet. Then they compared them, and they were able to use a model to adjust it for size, basically. And then they could create and apply a uniform gauge of brain connectivity, the number of synapses a message must cross to get from one to another in the brain. So it's not just how fast it's firing, it's how it moves across neurons.
Yeah.
And so our brain seems to be, you're saying, based on the same sort of prototype model that all of these animals' brains are based off?
So yeah, they're all the same.
Yeah. Okay.
Yeah. Basically, they're all the same.
They're all the same.
Very cool story.
It is a very cool story. And finally, for the quick stories, we were supposed to be quick.
Have these been the quick stories? I got on my... Okay, I'll talk I'll talk slower later.
Quick stories at the top. No.
Kiki, you're showing the run sheet still also. I don't know.
Oh, hey, everybody, look at the run sheet. Yeah. Don't you like it? It's pretty. You can see what we're doing. So, yes. We took a baby picture. We did. Scientists took a baby picture. Well, it's actually a distant image of for the first time ever, 2 planets orbiting a star. Very much like our sun, the 17 million year old star. Only 17 million years old. It's a baby star.
Beanie star.
Little
Hey, what's the
baby
first
star.
time ever? Is it that it's 2 planets or the first time we took a picture of this star?
That there's 2 planets
Just 2.
orbiting this star that we imaged. We took a picture. This star is called TYC 899876001, and it has 2 gas giants that are about 6 and 14 times the mass of Jupiter. And they exist about 300, they're very big, super Jupiters. They exist about 300 light years away, so, pretty far, but considering, 17 million years and 300 light years to get here, really not that big a deal.
Yeah, they'll be watching I Love Lucy in about 200 years.
They were observed using the SPHERE instrument on the Very Large Telescope from the Southern Hemisphere. The SPHERE instrument has a special light blocker that it blocks out the light from a star so that you can see everything else around it. And so it was imaged using this special coronal filter. The 2 planets orbit really far away from their host star, 320 and 160 astronomical units out. That's like 32 and 16 times the distance of Saturn from the sun. So it's like multiples of Saturn.
Well, they're big boys. They need space.
They're huge and they're way out there. But it's a very, very young system. And this is the first time that we have taken a picture of a baby sun with these little with their stars in tow. We have only previously been able to infer the existence of planets around stars through how they affect the wobble of the star through gravitational influences, or when planets occlude the light when they transit in front of a star, between us and a star. Mm-hmm.
This is some next level imagery going on.
Yes, it's very cool. Very, very cool. But it's an interstellar baby picture. We can show it to that star later and be like, look at when you were 17 million years old. You were so cute.
Except by then, we'll all be dead.
Oh, Blair, why do you gotta ruin it?
Bringing us down, man.
Actually, if we send it a picture now, won't it be like 600 million years old when it gets it? 617 million years old?
And 17 million and 600 years. If you just tuned in,
yeah, that's right.
You are in the midst of This Week in Science. If you're interested in supporting the show, you can head over to TWIS.org and click on the Zazzle link. The Zazzle link will take you to our store where you can find a mug, a shirt, mouse pad, or maybe even a face mask. So click on over there, browse our store, support TWIS. All right, it is time for our weekly COVID update. That was like like the least energetic COVID update. Boop,
I know, because I'm
ever.
like, oh no. That's why you have to end with I wish I had a trumpet and a music.
Sad trombone. Well, yes, numbers do keep rising. Globally, total numbers of cases are increasing at an accelerating rate. This indicates a lack of overall control of the situation. There are some countries that have gotten things under control. New Zealand, I'm looking at you. But many others are still in the midst of the COVID pandemic and not faring well. The United States is number 4 in per capita daily incidents behind Brazil, Bahrain, and Oman. Goldman Sachs estimates that a national U.S. mask mandate would increase mask use in public by 15%, which could reduce the need for more restrictive social distancing measures like closing businesses and save the economy $1 trillion.
This is what's so frustrating is the whole
Yeah.
reason we're back, right? Is the economy, the economy, the economy. If the decisions we were making were really about the economy, then someone would go, everybody, put on your mask.
Right? Yes, that's what we want to do. But everybody is not that. There are a lot of people who are, and we thank those of you who are being responsible and wearing a mask in public. Thank you very much for doing so. But there is the question about vaccines. What's going on in the vaccine front? Will we one day be able to walk mask-free through a shopping mall? I don't know, but that day will come if we can get a vaccine put together. And in the last week, there have been a few papers suggesting that progress is being made. The money and the effort going into vaccine production at this point in time is actually paying off. Multiple vaccines have stimulated immune responses and are headed now toward phase 3 clinical trials. There's currently 10,000 people taking the Oxford vaccine, which has been produced with a mutated adenovirus vector, and Moderna is looking to begin a trial of 30,000 people for its phase 3 trial. There's also a Pfizer vaccine and a couple of others, one in China that is another adenovector that is headed toward phase 3, all doing very well, having, when I say stimulating an immune response, what they've done is they've gotten the antibodies to perk up in the test subjects, although those test numbers, because they've been early phase trials, clinical trials, have not been huge. So what we know so far is in the people tested, there has been relatively safe efficacy. So an immune response that's showing up, antibodies that are growing appearing in the bloodstream, similar to the levels found after infection. Also, T-cell responses have been measured and detected in some of the vaccines as well, which is important for long-term viral immunity. Now, there was a question from our listener last week, Paul Riley. I punted to this week, and since there's been so much vaccine news this week, I wanted to answer the question from Paul. If a vaccine is just dead virus, then why is it so hard to create a COVID-19 vaccine? Well, vaccines are not just dead virus. Those are the very simple versions. We started with dead virus for diseases like smallpox, but I've done it again. Stop screen sharing. Thanks, Blair. No.
I didn't want to interrupt your flow.
My flow.
Yeah.
Here I go, yo. But through with the COVID-19 vaccine, you don't necessarily want to just use dead virus. Dead virus can have other negative reactions that you don't necessarily want. And sometimes the dead virus isn't necessarily completely dead and can get into cells anyway and cause actual infections. Polio was one vaccine in which that happened for a very long time. So what they do is they take little bits of the vaccine of the virus, and in the case of the SARS-CoV-2 virus, they are looking at things like the spike protein and taking those little pieces that the body recognizes and attaching them to another vector. So in the case when I said the adenovirus vector, what that is a in the case of the Oxford vaccine, I believe the adenovirus is it infects other primates, it's a cold virus that infects other primates, not humans. And so we shouldn't have an immune response to it because it should just infect the cells and give that little bit of the SARS-CoV-2 virus that we are introducing to our cells to the body to react to. And so the idea is that you try to get that little bit of the virus that you want protection for into the body so the body recognizes it and creates antibodies against it and continues to fight it off. Something that I found really interesting, a comment that a doctor made in talking about the difficulty of creating vaccines for respiratory viruses specifically, is that if you think about your lungs and your respiratory tract, you think of it as inside your body. But in reality, it is an extension of the outside of your body. It is epithelia. Epithelial cells are your skin. And if you've ever thought about, how hard it is to get things into your skin, you have to use a sharp needle. You have to use a knife. You have to do pokey things to get into the skin, right? You can't just, yes, we can. We have ways now to rub stuff on the skin and have your body absorb it, but it is not necessarily an easy process to get things through the epidermis, because the epidermis is meant to protect, and it does that in the respiratory system. And so in order to get protection, antibody protection within the body for a virus that affects you through the respiratory system, you have to you really have to do a workaround to be able to get the body to recognize it in the right way. So it's actually not as easy as some other viruses to create a vaccine for something like SARS-CoV-2 because it's respiratory.
And so we've what they're testing so far is the antibody levels based on these vaccines, right?
They're testing safety. So whether or not, do people have bad responses? Do they go, is there anything beyond a sore arm or a headache or a small fever?
Right. I more mean when they're deciding if something has efficacy, if it's an effective vaccine, they're looking for antibody levels. But we don't know. This has to be a long-term study because we don't know if they maintain antibodies or if they go away and then you can get infected by the coronavirus again.
Right. And so data so far suggests that antibodies, there's a new paper out that suggests antibodies at least stay in our system for 6 months. So after infection, they're suggesting you are at least going to be protected for 6 months. So the hope would be that
since we've been dealing with this for 6 months, does that mean that it's an ongoing thing? And so far for 6 months, or people have dropped off? I mean, that's
so that means worst case, you could have to get a booster every 6 months till this thing's gone.
Yeah.
So a couple notes here.
Worst case.
One is absolutely fascinating the way you described the lungs as epithelial. I've never thought of lungs this way.
It's connected to the outside world. It's out there.
The other thing is, if you take your lungs and actually spread out the material, it's this, what is it, the size of a tennis court?
Mm-hmm.
So you're talking about protecting your skin in a way, but it's the size of a tennis court, and you're just not aware of it. It's massive structure. Wow.
Yeah. And the other.
Go ahead.
Go ahead. The other thing, what?
Oh, the other thing I was hearing too, the other thing I was going to say was I've also heard reports that as they've been doing this increased testing and been able to test people who don't have the symptoms of the virus, that they're finding they're getting hits that are 10 times or much higher even than the reported cases.
Yeah, and so this is another interesting question. How much further spread within communities is the virus getting than we actually know about? Is it 10 times more? But still, even if it's 10 times more, that's still not a lot yet. And that's another aspect of the difficulty of testing a vaccine, is that you need to get volunteers. The best situation is to get volunteers from an area that's having an active outbreak for the test vaccines, because then the likelihood of that person interacting with somebody who has the virus and being exposed to that virus, and then actually testing whether or not this test vaccine does anything, is higher. So if you where you look for volunteers to test your vaccine is also very important. But then, are you getting a diverse enough sample to be able to really represent the full demographics of humanity to know, is it effective for everyone in the same way?
Yes.
Yeah,
Are you
so
only
that-
testing male mice?
Right? Yes. No, that's
Yeah.
not going to work. But there are a lot of difficulties to creating a vaccine that you don't necessarily think about at first. It's not just take a dead virus and put it out there. And other times you might create your vaccine with your little bit, the antigen, that little tiny bit of the virus that you're going to show to the immune system. And maybe it doesn't create a big immune response. And so then vaccines are mixed with things that are called adjuvants. And the adjuvants are in there because they shake up your immune system and force your immune system into waking up. And because they make your immune system wake up, it's more like your immune system's more likely to go, oh, what's this little bit of virus? Sure, fine. I'll have a response to that now. And so
so the antigen is like, this reminds me a little bit of there was this one village in Mexico, I think it was, it was South America at least, but I think Mexico, during the swine flu outbreak, that this one town had like the mortality rate was way lower. Everybody was sort of surviving it, and they ended up coming to the conclusion that because the well water in this town that everyone was drinking had an especially high arsenic content, that their immune systems were sort of primed all the time to be that they were under attack, and these people were generating more white blood cell response.
Right, and they probably die at a younger age because of all the arsenic, and they have all sorts of other health problems. So don't go drinking arsenic. But...
No. Oh, yeah, please, nobody take any medical advice from that scenario. But the idea is would that sort of be how the antigen is work? It's sort of priming the system to be on high alert, and then
the adjuvant primes. The adjuvant primes the immune system to be on alert, and then the antigen, which is the little bit of virus, is in there, and the immune system goes, oh, hey, you're not supposed to be here. Yeah. So,
cool.
So sometimes you have to figure out what you can mix the little bit of virus with to make the immune system respond in the way that you want. And you also have to, if you're using something like an adenovirus vector, you have to make sure that it is something. Adenoviruses are viruses that cause colds. They infect us very often. And so if you have a vaccine that is made with an adenovirus that is similar to something you've been infected with before, your body may have an immune response just to that adenovirus, but not to the virus that you're trying to create immunity for. So there are different balances and complications to how we create these things. The mRNA vaccines that are being created by Moderna, they're brand new, and so those are going to be really interesting because they, like a virus, use the machinery within your cells to create antibodies. Yeah. And so
cool.
All sorts of technologies and new ways of doing it, but everything is being tested, which is the most important part, and we do not want them to skimp on the testing. That's for sure. And anyone out there who is volunteering for any of these vaccine trials, thank you for doing that. Thank you so much for helping all of us.
Yes, and
move to
and
a
if you
vaccine
happen
faster.
to be the person of color, any form of minority, go out, find where if this is going on in your community, and volunteer for this, because the more represented you are in the study, the more effective the treatment will be for the greater population.
Yeah, studies are usually done on white men.
Well, but yeah, it's
sometimes women, but there is a male bias in a lot of medical tests.
Yes, absolutely. Yeah, the demographic of who's participating in the studies is going to determine the efficacy of the resulting drug on the greater population. So if one of these studies is going on in your area and you can contribute your diversity to it, please get involved.
And if anybody has any questions for This Week in Science questions, you can ask about COVID. You can ask about all sorts of other things, but you can email me, Kirsten at thisweekinscience.com, or leave us a message on our Facebook page. Sorry, I was distracted. My cat just fell out of the sky. All right. But moving on with the vaccine information, once we get a vaccine, that will be the interesting part. And our government is working very hard to try and get everybody access to vaccines once a vaccine is available. New news, the U.S. has agreed to pay $1.95 billion to Pfizer Inc. and BioNTech SE to secure 100 million doses of their experimental COVID-19 vaccine for Americans for free of charge. Now, America has a lot more than 100 million people, so hopefully there will be more vaccines available.
Yeah.
You know, who's going to get those vaccines?
Who do you think?
The National Academies of Sciences, Engineering, Medicine, and the National Academy of Medicine have formed a committee that will develop an overarching framework to assist policymakers in the U.S. and global health communities in planning for equitable allocation of vaccines against COVID-19. So there's a committee.
Okay, well, people with healthcare are going to get it first. So that's a whole thing. When we find one that works, the recipe should be free.
Yeah. There's another study that came out this week looking at hydroxychloroquine. It doesn't work. It doesn't work. Another study was published.
That's not true. Hang on. It works fantastically.
Not for
it's probably one of the best drugs.
infection.
Oh, no, not for that. No, that's totally wrong. Yeah, it's horrible for COVID.
Horrible for COVID, yes. For in use against SARS-CoV-2 infection in non-human primates. They used hydroxychloroquine to try and help monkeys that were already infected to see if it would reduce time of infection or ameliorate the symptoms. It didn't help. They also tried to use hydroxychloroquine in these monkeys to see if it would help keep them from getting sick, and it did not help. So it's just another study adding to the pile of evidence that hydroxychloroquine is not really a useful drug in the sense for COVID-19.
Let the people who have prescriptions for it use it instead.
Yes.
Yeah.
Don't stockpile. Let the people who need it use it. Yes. Hey, this is This Week in Science. Thank you for listening to TWIS. You're the reason that we are able to do what we do every week, bringing you up to date and down to earth news and views on science. And with your help, we can do even more. We can continue to bring people our sane perspective. I think we're sane, kind of. Our perspective to a world full of misinformation. Head over to twist.org right now. Right now. And click on the Patreon link and choose your level of support. Be a part of bringing sanity and science to more people right now.
Do it. Come on. No, wait. No, we're waiting for you. We're waiting for you. We're not going on until you click the buttons.
No, I'm going to go on. Let's go on.
Okay. All right. Never mind.
All right. And Justin, do you have any stories?
Oh. Yes. Oh, I do here. As soon as it loads, switch to state. Okay, here we go. So this is a good story. This is stone tools have moved back the arrival of humans in the Americas by thousands and thousands of years. Again, findings of stone tools move back first immigration of humans to America by 15,000 years. Currently, we're hovering at around the 15,000 year mark for the sort of generally accepted time. This is revealed in a new international study from the University of Copenhagen, where researchers have analyzed ancient material from a Mexican mountain cave. And they're calling it proof that the first humans arrived in America at the very least 30,000 years ago. Probably more like 30,000, 33,000 or something like this.
No. It was 15,000.
It was 15,000. Well, or was it? So here's the quoting. Okay, quoting the press release. Approximately 15,000 before science was hitherto able to render it probable. First, couple of problems with this. First, there's no need to bring back the word hitherto. There might even be some danger in it. It's gone for reasons. We kicked it out. It's maybe even a little bit dangerous. Okay, so creeping the case. Okay, so that's the first problem. Second problem is French-Canadian archaeologist Jacques Singmars, who we've talked about on this show before, and he had a Yukon discovery 30-something years ago in the Bluefish Caves, where he found animal bones worked over by tools that dated 24,000 years old, which by itself renders this date completely probable. So this was Jacques Singmars. He had his discovery. He went around and tried to tell people about it. He was literally laughed at for suggesting a timeline older than 13,000-year-old Clovis sites. Jacques Singmars was right then and is still right, it seems, despite so many claiming to have found the first evidence of older than Clovis over and over again throughout South America. Okay, still, this new study published in the journal Nature is awesome. The team of archaeologists and DNA experts from the University of Copenhagen, as well as universities in Mexico, UK, US, and Brazil, and others, all collaborated on this. The article, this is a quotey voice of Eske Willerslev. The article in Nature is a scientific hand grenade. The fact that it moves back the time of early immigration to America significantly is guaranteed to ignite a heated debate. Maybe. Eske Willerslev and his two colleagues, Associate Professor Michael Winter-Peterson, Assistant Professor Martin Sikora, made up a Danish contribution to the international team of researchers. The three scientists have conducted the DNA analysis of ancient remains from animal and plant material found during the excavations of the Chiquiwi Cave in northern Mexico. They found 1,900-ish stone tools. That is an amazing collection of tools from any site, and they dated them as far back as 30,000 years. And they also say that these they had already developed techniques for producing these tools that were sort of unique to the Americas. Sort of interesting too is that they describe this cave as also sort of having an impression that has 3 meters, I guess, of compacted earth, and in a sort of 6 layers of detritus and dust and all, a 10-foot column of ancient remains, which is so compressed and stable that using various advanced measuring methods, it has been possible to date the layers one by one from top to bottom. So also,
wow,
aside from all of the tools, a perfect column, if you will, of data has been stored within this cave. Each layer has contained deposits of stone tools such as knives, scrapers, arrowheads, which the researchers have also been able to date. The cave finds are extremely interesting. These archaeological finds are so far the oldest in America, and the excavated stone tools are of a type unique to America, says Professor Universidad Autónoma de Zacatecas in Mexico, Cyprian F. Andrelan, saying. Until now, science has assumed, that the earliest immigration in America took place approximately 15,000 years ago. There's also sites, there's one in Pennsylvania, which has been dated at 19,000. But again, it's out of the range. Can't be true. You're going to have to look at this one. At the time, there were no other. Okay, so at the time, they've been assuming this because of the last glacial maximum that sort of covered North America. And they have always assumed that until that broke up, nobody could get through. So at the time, 30,000, so at the time, there's this narrow opening, though, that happens about 15,000 years ago, which makes it possible to walk from Siberia onto the American continent and then later down through. That's been why. It's been part of why the theory has been there, because you can look at the glacial thinning, and it kind of matches up with some of the more predominantly accepted sites, the Clovis sites, and some of the others now in South America. This is my side note to the story. The crossing area that they describe would have actually formed a chain of islands much, much earlier, which we've talked about that in a story on the show. And these islands would have been able to be navigated by a boat, which would have also predated the glacial maximum around 19 to 26,000 years ago and allowed for pleasant enough boat travel down the coast. So if you were boat faring 30,000 years, 33,000 years ago, you wouldn't have that massive glacial cap, but you would have this sort of chain of islands showing up between Siberia and Alaska that you could sort of hopscotch through, which if you have little islands popping up in the sea, you might have something to hunt, something to hunt on these little islands all the way across. And then you would have had a coast without a glacial maximum. So 30,000 years ago, when the first stone tools were left in the Chiquiwi cave, the ice cap had not yet covered all of North America, which means it would have been possible even to walk down from the North American portion of the continent, says Eska Willerslev.
That would have been, it would have been a long walk. But the one thing that I'm thinking about in this whole story, though, is DNA. They found DNA in the cave. I'm going to guess, yes, some of it was dead animals, but part of it is probably also fecal remains. I'm going to guess that part of that DNA is like what animal, like if there's black bear DNA, it's what was the black bear eating? Probably plants and berries and, how are you finding it there?
So that's a good guess. They found black bear, a wide range of rodents, several types of bats, as well as sparrow and falcon.
Yeah. And it's going to all be in the layers there. Yeah.
It's all in the layers. And they also were able, it also gives them, because of the different plant compositions changing over time from these different steps, it also gives them sort of climate change information, as well as the sort of biome of where these caves are located over time. It actually is a huge treasure trove of information that will be sorted through and written about and studied for decades to come.
Yeah, but, environment, ecology, climate, like all those things tied together, not just, hey, people, tools 30,000 years ago.
And so many tools, though. So many. I mean, that is amazing. That is just absolutely a dream come true to have all of that to study.
So interesting, though. There's so many tools, but no human remains. That they're not actually, there's no other. I mean, the tools have withstood the test of time, but you'd think it's somewhere nearby. Maybe not there, but there've got to be burial sites or people remains somewhere nearby.
Yeah, so that is very interesting, right? You don't, hey, I mean, I don't know what your house looks like. I don't have anybody buried where I'm living.
Yeah, that's true.
There was this practice in some of the early Middle Eastern, I think it was, where ancestors would be buried underneath the home or hut. You would sort of carve out an area under, and they would actually be buried underground in the house, which is probably where the idea for burial came from. Like, well, if they're going to be in the house, let's make sure they're under.
Let's make sure they're covered with a lot of dirt.
But yeah, for the most part, that's a very odd thing to happen. And conversely, when we look at the Homo naledi, the massive sort of
the cave
human
of bones,
remain,
right?
Yeah, the cave of bones. It's cave of bones, not a cave of tools. So right. Where's all their tools, or where were they? Where's all their food stuff? It's not there. Why?
And
they
so maybe
just apparently put the dead in one cave and maybe lived in another.
Yeah, and if people at this point in time were, smallish bands of people, they're stopped there for a small period of time, seasonally, maybe even. Mm-hmm. Maybe they came and went numerous times. Maybe they had a few different areas they frequented. We don't know what these people were doing, and so maybe they did use the cave to live in for a short period of time and then moved on to other places where they died and buried their people. Justin, you have more news. Tell me about the plants.
Oh, I did bring one more story. That's unfortunate. Yeah. I forgot all about it. Okay, so this is water plants are a nuisance for streams and rivers, nefariously blocking the flow and taking up volume. When they take up volume, they can lead to more frequent flooding. And so we always remove all of this plant material from streams and riverways to make sure that the water flow is more manageable. Mm-hmm. Well, now research by Royal Netherlands Institute for Sea Research scientist Loretta Cornacchia, published in Proceedings of the Royal Society B, in cooperation with Utrecht University, shows that vegetation in streams can actually buffer water levels. And by adjusting vegetation cover, they actually are more efficient at managing the flow. Says Loretta Cornacchia, they provide a natural buffer against hydrological changes. The information for water management are huge. The current strategy of plant removal does not only threaten a naturally balanced water level, it also threatens extremes biodiversity. The inland waters are freshwater hotspots for biodiversity. Vegetation is the guardian of this. So it's providing habitats and food for invertebrates, fish, helps stabilize riverbanks, so there's less erosion, removes excess nutrients from the water, so the water is more clean, I guess. Mm-hmm. At high flows, they find the vegetation is pushed to the side. It can even be dislodged to the sides, which then forms an open lane, sort of a fast lane down the middle for high flow traffic of water. During periods of low flows, aquatic plants actually gradually clog up the stream, slow down the runoff, and thereby prevent the water from draining too quickly. And this is the so-called self-organization process counteracts the variation in water flow that streams experience through the seasons. Last quotey thing from Loretta Cornacchia. I'm saying it very Japanese. I'm saying it very Japanese, but it's not a Japanese last name. Loretta Cornacchia, maybe. I don't know. The more nature-based approach to the control of water levels harnesses natural processes rather than the human control to maintain biodiversity hotspots. Insight in self-organizing plants and understanding their role in ecosystem resilience is essential to a future governed by global change, which, of course, the future might have some more rain. We might be blessed with a little more rain.
Not just a little bit more rain, but rain in atmospheric rivers. Rain that comes once every long period of time, causing these floods, and in which we would really having natural controls to flooding and water distribution would be great.
Yeah, especially when the water's up to the cows' knees and the chickens are sleeping in willow trees.
Especially. All right, I have some
Johnny
science.
Craft.
I know. I'm going to talk about brains right now.
Oh, more brains. Brains.
Because I do love the brains. So, we've talked before about the benefits of young blood to old brains.
Yes.
Yes. We've also talked previously about the benefit of exercise to brains in general. Exercise is good for your brain. And we know that there are factors in the blood that come from exercise that can potentially change the brain. So researchers publishing in Science this week, they took old mice and split them up into two groups. Now, old mice are 18 months old. That's not old in human years, but in mouses' years, that's old. And then they took half of those mice and they said, you get a running wheel, and you get a running wheel, and you get a running wheel. And they got to run and exercise. And the other half of the mice, they said,
you get a couch, and you get a couch, and you get a couch.
You got it. You got it. You saw where I was going. So they had sedentary old mice and active old mice. And they found that when they took the plasma, the blood plasma from the mice who were old and who had exercised and put it into the sedentary mice, it led to improvements in cognitive tasks related to memory, and it led to improvements in the liver and also in other aspects of metabolism. They were able to identify a compound called I said it a number of times before I actually did the show to see if I could say it, and now I stumbled. Okay, I'll try again. They called it glycosyl phosphatidyl inositol specific phospholipase D1. It's a factor.
Woo!
Yeah, it's a liver factor.
Awesome.
A liver factor.
I'd get in about halfway through and change the ending.
I know. You can also call it GLP, but this is an enzyme that splits glycosylphosphatidyl. Anyway, it's a thing. It's a factor that's an enzyme that splits other factors and it's in the plasma. It doesn't cross the blood-brain barrier itself, but they took this one little enzyme, and they determined that enzyme on its own could lead to changes in the brain of older mice. So the study looking at this glycosyl phosphatidyl inositol specific phospholipase D1, otherwise known as GPLD1, that they're now going to start looking at this as a way in which blood factors can actually transfer the benefits of exercise to the aged. One of the ideas being that some people are old and frail and unable to exercise, and so they cannot get the benefits of exercise to the brain, and so their decline mentally is much more rapid than people who are able to exercise.
So give them young blood.
But you don't have to. You don't have to now. Now we are looking for factors like this GLPD1 that can potentially be given. It improves liver function and leads to a bunch of downstream effects that could hopefully help improve brain function.
This seems like unnecessary extra work. Just give them the young blood.
Just give them. Why don't we just do that?
Hey, Coach, I got a question. How come every team in our league is sponsored by Plasma Inc.? Yeah, funny thing, son. So it's also part of why we all give blood donations before every game. I thought we were doing that for the good of the community. Well, we are. And it's also why we haven't won a game in a while. Well, nobody's really. They have to call all the games because everybody's listless by the fifth inning.
But
But
yeah.
yeah, I think we could just young people blood seemed like when we first were encountering this, I was like, oh, there's going to be this just draconian black market blood
For
market.
blood from the young kids. Yes.
Ah,
Blood from
Give
the
me
youth.
that youth blood.
there's going to be poor children in communities around the world whose families are subsisting on the young giving blood that goes off to billionaires in other third world countries. But it would be nice if we could synthesize a way around the whole leeching the young part of.
Yep. So looking
Make
for
us
those
a
factors
slightly
in
less
the blood.
vampiric society.
slightly less. Yeah. Yeah,
Still
but
capitalism.
it also identifies this liver to brain axis and suggests also that there are aspects of liver health that are important as we age for maintaining the proper factors getting into the blood to maintain the health of the brain. So this is a new area of research, very exciting. Yes, maybe we will not have to suck the youth dry. But for people who, moving on to another story that I thought was promising and exciting, people who have limb amputations and need prostheses, they either complain of having phantom limb syndrome, where they feel things that aren't there because their limb is not there anymore. Or they feel nothing at all because the limb is not there. And if they're using a prosthetic, there's no sensation in that prosthetic. So they go and they reach for things, they try and do things with the prosthesis, and they're unable to feel. So there's no feedback as to the pressure that they're putting on a cup or when they're touching something. Everything has to be visually confirmed.
This is something I haven't lost any limbs, but I do have lack of feeling in parts of my hand. And I have dropped and shattered at least 5 mugs because I don't know how much pressure I'm putting on the cup. And it drops right out of my hand.
Yeah. And so this new study out of let's see, where is this from? The University of Pennsylvania Medical Center. Oh, sorry, University of Pittsburgh Schools of Health Sciences. They've published in eLife their new study in which they're repurposing these new spinal cord implants. The spinal cord implants are used to treat chronic pain, and they just sit there, electrodes that are run into the spinal cord, and usually for chronic pain, they short circuit the pain pathways so that people don't feel the chronic pain as much or don't feel it anymore. But knowing this and knowing that sensory information is sent through the nerve trunks in the spinal cord, the researchers implanted electrodes into the volunteers' backs in their spinal cord, and then checked to see if they could trigger a feeling in the limb that was no longer there. And that's what they were able to do. And so the hope is that eventually they will be able to tie this with a prosthetic limb in the future, so that if somebody touches the prosthetic limb, if they reach out to grab something with the prosthetic limb, that they will be able to have sensation on from the prosthetic device that they actually feel.
So yeah, it's all electrical signals.
It's all electrical signals, and they did this.
And the target is there's that narrow strip on the side of the brain. There's something there for hand. There's something there for arm. There's a body location little map that the sensory information is going to. And what happens to folks is when they lose a limb, there's this little part of the brain that's no longer getting signals. It can actually get taken over by neighboring signal areas of the brain, or it can just keep screaming out like, hey, something's wrong, and that's where you get these phantom pains. It's this part of the brain that's not getting its signals anymore. And if you could find the pathway to give it signals, yeah, why not? You should be able to integrate this into, these robotic, feely, touchy, soft, fingery things that we're always seeing come across, like new, more fleshy, like robot hand, with touch sense. Those electrical signals, if they're diverted to this little patch of brain, should effectively give that sensation, that signaling pathway back to the brain, and you can feel again.
Yeah, so they are hoping that they'll be able to design spinal stimulators that can be fully implanted as opposed to connecting to a device that's outside of the body, which is what they did for this particular trial, and then demonstrate that the feedback between a prosthetic and the body can actually improve control of a prosthetic hand. Right now, they're just showing it's like proof of concept. We can make this, we can show that there is this matching of sensation, simulated sensation. But and the researchers say mostly we wanted to demonstrate the possibility that something like this could work. And I think it's a really promising direction, and I think very exciting for people who need prosthetic limbs or who want an extra limb, if you want to figure that out.
Wait, but the brain wouldn't have...
Oh, you could teach your brain. Yes,
really?
You could. For sure. Yes.
I'd take a third arm. Why the heck not?
Why not?
The question is, do you put it right out the middle or under one of the other arms? That's really the question.
In the middle of your back?
No, because then you couldn't lay down or sit. That's silly, Kiki. Don't be silly.
Sorry. Okay.
You would have to go middle of the chest, or I think...
No. I sleep on my stomach. It couldn't go there. Yeah, it would have to go on the side.
Under one of your arms.
It'd be in your armpits.
Then you'd have
really
to make
those anymore.
Instead of
need
t-shirts.
Armpit. Yeah, just have you'd be like a dragonfly with
Oh, you're doubling it, huh?
I'm doubling. I'm going under. I'm going to stay. I'm going to keep. I'm absolutely going to stay.
You like that bilateral symmetry.
I do. I like the symmetry.
Yeah.
But under each armpit, because
then Justin, I figured it out. Right out of the top of your head. Just an arm right out of the top of your head.
Right out of the top. I think we have made it to one of our favorite times in the show. This is This Week in Science. Do you want to help TWIS grow? Get a friend to subscribe today. All right, it is time for...
As you're pulling that up in the YouTube chat room, Eric Combs, happy birthday!
Happy birthday!
I do, yes. I get a list of all the birthdays of everybody who logs into the chat room on YouTube. It just auto-populates. Eric Combs, happy birthday. Yeah, it's the new data capture thing I added to the... You didn't know about... Well, of course you didn't. I hacked into the... It's okay, it's fine. Happy birthday, Eric. I'm sure he's happy that I said happy birthday. It might be weird that I know it's his birthday, but it's happy birthday.
Fantastic. It's time for Blair's Animal Corner.
With Blair.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
You all know that coworker. You know the one. The one who's really outspoken, kind of a little bit aggressive, really likes to get their way, and often asserts themselves as the leader. Well, that particular type of person also exists in fish society. And a new study
there's a fish Karen?
Yeah. Karen, I don't know if Karen's dominant. Karen's just annoying. I feel like this is like a... I don't know, like a Greg or something. Like, Greg's telling us what to do again. But so this happens in fish too. And a new study on fish behavior shows that dominant individuals can influence a group through force. That can be physical force, or in Jeff's case, verbal force. Or in other cases, passive individuals are actually better at bringing a group to consensus. So the difference between these two scenarios is actually quite interesting. This comes from Max Planck Institute of Animal Behavior, the University of Constance, and the University of Texas at Austin. Those three, I couldn't say the word Austin. That's funny. But it kind of shines light on the potential of domineering individuals to actually obstruct or hamper effective communication in organizations. So depending on the situation, you might not want somebody that's take charge. And so in order to figure this out, they studied cichlid fish. They looked at them because cichlids have strict social hierarchies. And there's usually a dominant male that controls resources, territory, and space, or many dominant males, depending on the size of the group. The dominant males are usually more colorful. They're usually more aggressive. And they are central in social networks. And as I mentioned, they control resources. So they are considered to be very influential. Drab subordinate males, they are passive, non-territorial, and have little or no control over resources. But they wanted to see if they wielded any influence in that society. So they examined how information flows between dominant or subordinate males in the groups in two different contexts. One was routine social behavior. So just like eating, swimming. What else do fish do, really? And then more complex social learning tasks. In the complex learning task, dominant or subordinate fish were trained to a certain colored light on one side of the tank to mean that food would arrive at that location. So a nice red light or something says food's going to be dropped right here. So then those informed individuals were then brought back to a new group of individuals that had not been informed of that information. And then researchers looked at which group more quickly learned to associate colored light with food. So who's able to communicate to the group at large? Come over here for food. So what they found is in the routine social interactions, eating and swimming, the dominant males had the greatest influence on the other fish by chasing and pushing the group around. But in the complex task where they were asked to communicate this kind of idea that this light meant food to the group, it was actually the subordinate males who had the greatest influence on their social group. Yeah, so what does this mean? So you can kind of extrapolate this and think about how there's an opportunity between kind of how that relationship affects the other fish and who is a reliable messenger. So what they're kind of the extrapolation here is that animal or human societies have individuals that are in positions of power or prowess. And often those things are associated with aggression, intimidation, and coercion. So not that dissimilar so far, right? But communication requires a diversity of voices. If there's one person yelling, it can often turn people off. And so these results from a natural system with fish show that alternative pathways to positions of power are actually useful in creating a stronger advisory, governmental, and educational structure. So this is
all right,
kind of
Poindexter. What's that light mean? Well, actually, sir, it means probably going to be food dropped over there within a very short period of time. All right, let's get a move on. Let's go over there. Hey, finally make yourself useful.
Yeah, so I think that's
very
kind of the
bullet,
idea, right? Is that you need different types of influencers.
Yeah.
And so it's fish. I'm not going to tell you exactly what this means for humans, but what I see here is does our society, I'm just posing the question, does our society prioritize certain types of voices being put into decision-making roles?
Absolutely. Yes.
Well,
And is that right?
Yes.
Or should we have more of a diversity of people
So-
giving those sorts of recommendations to the general public?
Here's the thing. Would you say there's a correlation with people who are really good at handling large amounts of information but lack social skills versus people who are very good with their social skills but maybe don't pay attention to any of the details?
Ah, there
It
you
might
go.
It
The
might be
sweet
actually
spot
not
is
different
where
than fish.
Yeah, the sweet spot is where you have somebody who does well in both domains. But yeah,
Freaks.
Yeah.
That's
Well,
what we
and I
call
mean,
them,
I
freaks.
just think about, the coronavirus briefings here in the United
Justin.
States, where we had one person talking who was definitely dominant, but maybe didn't have as much information. And then you had another person talking who had a lot of information, but had to stand back and roll his eyes. And so who actually would have been the better person to listen to in this situation, and who maybe was listened to more by the general public and trusted more according to polls?
Yep.
It's interesting.
It is very interesting.
This
This is making me think. Yeah, this is making me think of another study that came out of the Santa Fe
Correlations are real, by the way.
Institute this week.
Yeah.
That they're looking
Real thing.
at monkeys. I think might have been macaque monkeys, but groups of monkeys. And the take-home message from the study is that groups of monkeys are better informed than the individuals within that group, and that the groups are like little computers. That the monk that they.
Oh, I did see that one. Yes, yeah.
Where they they looked at these monkeys as they were forming groups and leaving the groups while they were foraging in these fruit trees. And so the groups were kind of the signal of whether a tree was good to be foraging in and how good it was. But there were decisions made by the individuals to leave a group and make another group, or to join a different group. And so there was this constant flux. And the big message from it is that individuals don't necessarily understand or have they have the components of making decisions, but it's really the group knowledge, that emergent property of the group, that is actually more intelligent. And this is kind of like the wisdom of the crowds. But the other side of it is that the tragedy of commons. But then there's also they looked at the fruit trees themselves and determined that these monkey computers were very inefficient, and they could have been using the fruit trees much more efficiently if they had done it differently. So there was wisdom in the group, but not total dominance over that knowledge. Like it didn't make them super efficient and perfect. There were still inefficiencies.
Right. So I think this is where so a group has a greater total
According
knowledge.
to who?
But when you then have to act on that, sometimes inaction is the result, or disorganized action. So often you need a leading voice, which in this case with the cichlids, you had one fish that knew something that no one else knew, and they had to take the initiative to kind of show everybody. So a leader still had to emerge. And so that's why, having diversity of understanding and background knowledge is so important, but also there are times when people need to stand up and speak. And it turns out it doesn't always have to be the same kind of person.
Right.
Yeah,
but
the unfortunate thing, though, is in your scenario, somebody pulls the Excalibur out of the stone, and somebody standing right next to him, Mordamus, like, "Oh, thank you. I am now the King of England. Thank you very much."
But then it makes me think of the rat study from last week, where it was like, well, the rats and people don't always do what is quote unquote right. Who is going to step up if nobody's stepping up?
Exactly. So there's actually one of my favorite TED Talks I've ever seen, not to go totally crazy on a tangent, is that actually the initial leader is not the most important person in making a movement. It's the first follower.
Ooh.
Because you can go and do something crazy and start talking on your own, and you're just one crazy person. But to follow that crazy person is actually putting yourself out there in a big way. And then for every additional person that's following, it becomes less risky. But the risk is actually highest for the first follower. So it's interesting that everybody has an important role to play. You don't have to be the change maker, but starting a support system is often more important. I think it's very interesting.
Mm-hmm.
Can't have a pyramid scheme without
What?
you're
No
just one
pyramid
person.
schemes.
No.
Wait, what?
No pyramid schemes. No.
No, that's what all power is.
Yeah.
It's
Well,
all
speaking
forms of power.
conspiracies, let's talk about invasive species.
Invasive
There's
an
hedgehogs,
invasive
my
species
metal
out there?
band.
So invasive species, in particular, this one is looking at hedgehogs and ferrets, which are animals that I talk about through my day job all the time because they're illegal to have as pets here in California and in Oregon, and in a lot of the United States because they're not from the United States, and they have a huge potential of becoming invasive in any place they go that is not where they're from. Hedgehogs because they are so good at protecting themselves, and ferrets because they are amazing little hunters. So all of that together means they have a potential to wreak havoc on an ecosystem that is not theirs. And because people have them as pets, this is often why they're released into the wild. Whenever an animal is a pet, the likelihood they could escape or be released is high. People don't understand this animal isn't from here and they just let it go in their backyard. And it can cause a huge problem on the ecosystem. And so how do we either eradicate these invasive species or reduce their negative impact on an ecosystem? And so this was looking at ferrets and hedgehogs in New Zealand, a place where they are invasive as well. What isn't invasive in New Zealand? Sometimes it feels like there's more invasive animals than local animals. But ultimately, because they're an island, they're especially susceptible to these problems. And they were trying to figure out how these animals hunt, and specifically how they find and target prey, to see if they could use that information to reduce their impact on the ecosystem. And this is also based on the idea that both of these animals are nocturnal. Both of them are extremely reliant on their sense of smell. And previous research has already shown that chemical camouflage could be effective in deterring other invasive species from harming vulnerable bird populations. And chemical camouflage is when you basically just spray a whole bunch of bird nest odor all over a space before anybody lays any eggs. So then they keep going, is that fresh eggs? And they walk over and they're going, oh, there's nothing here. And they do that for like a month before the eggs even show up. So then by the time the eggs show up, they're kind of jaded and they're over it. They're done looking for that stuff. Like, that's a false signal. So the chemical camouflage has been used in the past. And so the researchers wanted to see if and how chemical camouflage could be used to protect birds from ferrets and hedgehogs. So they found how these animals were using their sense of smell was really the crux. So they are using their sense of smell to locate prey. For ferrets, they're eating things like rabbits and large game. For hedgehogs, mostly they're eating insects, but they're both opportunistic. So if they find a free meal in the form of an egg or a baby chick, they're going to take it.
Right.
Wouldn't anyone, right?
I would.
So if you found a free meal, no strings attached, you would take it.
Meal.
Yeah.
Yes.
So how did they do this? They captured local ferrets and hedgehogs, and they ran them through a series of controlled treatments in outdoor enclosures. They resembled natural habitats, and they wanted to see how they categorize smells. So whether they group smells into types of birds or how they habituate to smells, how do they process this input? And so from that, they found that their hunting strategies and their priorities were specific and very easily manipulated. So it looks like ferrets generalized all avian smells. Avian smells. I said that
They
right.
really
Yes.
want those eggs.
So basically just like any bird smells great. Hedgehogs were much more specific. So they were really looking at maybe ground nesting bird eggs that are around a specific type of year. So it was kind of more specific.
It makes sense. I mean, hedgehogs they're not climbing trees.
No, and they are seasonal. So hedgehogs,
they
I know
hibernate.
this for African hedgehogs. I'm not 100% sure about the European ones, which I think are the ones in New Zealand. But African hedgehogs estivate. So they go through summer hibernation.
Right.
So they are extremely seasonal. So it would make sense that they'd only be smelling for animals that lay eggs when they are awake. So that would make sense. So based on all of this, they think that they will be able to protect bird colonies by distributing specific smells around an area, saturating it, and therefore doing this chemical camouflage for hedgehogs and ferrets. They become accustomed to the smell, they ignore it, they seek out other prey.
So no birds for you.
Yeah, and this also
the birds.
It's general enough that it might be really hard to synthesize or capture a ground quail smell that is, a protected species. But it seems like they can be much more general. They could put out chicken smell and it would be enough.
At least for the ferrets.
Mm-hmm.
Yeah.
Definitely.
Hedgehogs might be a little bit more difficult.
Perhaps.
I don't know. It's so funny. When I think of hedgehogs, I mean, I guess, being invasive, being a pet species, that totally makes sense. But at the same time, I really do not think of hedgehogs as this incredibly efficient hunter and survivalist in the wild. I think of hedgehogs as these little cute little things. Ferrets, on the other hand, they're going to get in there. They're wily. They're predators.
They can hunt full-grown rabbits. Yeah.
Yeah. So ferrets, I'm like, yes, I'm down with, but the hedgehogs, I'm having some mental gymnastics to make this work.
They're not hunting. They'll eat an egg. Is really what it's about.
If I came across an egg, I'm going to eat it.
But I mean, here in California, that's not even the main concern. The main concern is that we have indigenous small mammals that live underground, and hedgehogs live underground, but they're terrible diggers. So they actually steal other animals.
Yep.
So they go underground, stick out all of their quills, and get out. This is my house now, and that pushes those animals up. Where there are hawks and snakes and other animals waiting. Exactly.
You know who loves hedgehogs?
Who?
Hawks.
Yeah. It's because they pick them up and
really
drop
well
them,
when there's hedgehogs around.
Thanks, hedgehogs. Caw.
Yeah.
Oh my goodness. Well, I think that we have come to the end of another show. But I did need to answer the question from the title of our show, which for this episode was why the Earth isn't flat. And apparently, according to a study this week from the University of Pennsylvania, it's because it's made of cubes. It is. Publishing and Proceedings of the National Academy of Sciences, a team of researchers from the University of Pennsylvania, Budapest University of Technology and Economics, and the University of Debrecen. They looked at a whole bunch of little tiny rocks. They broke rocks, they measured rocks, and they used mathematical models, geology, and physics to determine that Plato's conjecture that the idea of matter, everything is composed of the smallest indivisible object is something of a cube. Well, he might have been onto something. Apparently, measuring all the rocks on the planet based on the way that geological forces in 3 dimensions grind up rocks and break them, the random fragmentation that happens again and again and again leads to shapes that are generally cubic. And so the average shape of indivisible rock on our planet it's cubes. If you go look at nature, they've been eroded and made soft at the edges.
Way too much Minecraft. I've been in nature. I've collected rocks.
Yeah, I'm having trouble with this one.
They're
Earth is cubes.
Flat little.
Is this like imagine a spherical cow? Are we just trying to make it easier?
Yes.
Just
no, I'm not buying it.
Well, it would make some things a little bit easier. What they say is they think they have found what appears to be a fundamental rule of nature, but it could help identify patterns that lead to rockfall hazards, likelihood and location of fluid flows, oil or water in rocks, and it could help with a lot of identification of geological features and things that happen geologically. So anyway, the researcher Dr. Gerald Mack says, when you pick up a rock in nature, it's not a perfect cube, but each one is a kind of statistical shadow of a cube.
It's the spherical cow. That's what that means. Oh my God.
You can. Have you ever seen rock art where people make shapes with different rocks? Like you can make any shape out of a bunch of discombobulated rocks.
I'm
Perfect.
just gonna. Hey, from now on, I am Team Cube Earth. I'm just saying.
Okay.
I do still stand on a corner then, Kiki.
I do still find it very fascinating. I do still find it very fascinating how quickly this flat Earth thing has gone around the globe.
Oh, around. Yes. Very quickly. Thanks to the connectivity of the internet. It spins us round and round and round.
Also, remember, the moon is round, but Earth is not.
Yeah.
Well, I think we can still be round, but we're supposed to be flat, and you can foul off all the sides.
Yeah. No, all the other planets were spheres. All the other planets are spheres.
No, we're not in the snow globe.
We're not.
Thank you for listening. I hope you enjoyed the show. Shout outs to the friends of the show who help keep things running. Fada, thank you for your help on social media and in the show notes. Gord, thank you for manning the chat room. Identity 4, thank you for recording the show. And I want to thank our Patreon sponsors and the Burroughs Welcome Fund for their generous support. Thank you to Eric Combs, Flying Out, Guillaume, John Lee, Ben Rothig, Ali Coffin, Maddie Perrin, Gaurav Sharma, Taylor P.S., Josiah Zahner, Sarah Forfar, Donald Mundis, Rodney Lewis, Stephen Alberon, John Ratnaswamy, Dave Friedel, Daryl Myshak, Stu Pollock, Andrew Swanson, Corinne Benton, Sky Luke, Paul Ronovich, Ben Bignell, Kevin Reardon, Noodles, Jack, Sarah Chavez, Paul, Jason Olds, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina, Sue Doster, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, Ken Hayes, Howard Tan, Christopher Rappin, Richard, Brendan Minish, Melazond, John Gridley, Richard Porter, Christopher Dreyer, Mark Mazzaros, Artyom, Greg Briggs, John Atwood, Robert, Rudy Garcia, Dave Wilkinson, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Alex Wilson, Dave Neighbor, Costi Ranke, Matthew Litwin, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Disney, Patrick Pecoraro, Gary S., Ed Dyer, Tony Steele, Ulysses Adkins, Brian Codron, and Jason Roberts. Thank you for all of your support on Patreon. And if you are interested in supporting us, you can find information at patreon.com/thisweekinscience or click the Patreon link at our website. On next week's show,
we will be back on Wednesday at 8 p.m. broadcasting live from our YouTube and Facebook channels and from twist.org/live. Is it supposed to be yelled? It's all in caps. You said the...
Yes.
Want to listen to us as a podcast? Just search for This Week's in Science wherever your podcasts are found. If you enjoyed the show, get your friends to subscribe too.
For more information on anything you've heard here today, show notes and links to stories will be available on our website, www.twis.org. And you can also sign up to hear our newsletter.
There's
You
one
can
coming soon.
Yeah,
Ooh,
it's true.
nice.
You can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in that subject line, or hitherto, forever. Your email will be spam filtered into oblivion.
Oh, no. Can't bring the word back. It's so awful. You can also hit us up on the Twitter, where we are at Twist Science, at Dr. Kiki, at Jacksonfly, and at Blair's Hitherto. We love your feedback. If there's a topic you'd like to discover, address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
It's Blair's Menagerie. We'll be back here next week, and we hope you'll join us again for more hitherto unheard great science news.
Oh, and if hitherto you've learned anything from the show, remember,
It's hitherto all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth, and I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.